Have you ever kept an old high school notebook just like tucked away in a box somewhere in the attic, just in case you ever needed to remember geometry?
Oh, yeah. Like you're suddenly going to need to calculate the area of a rhombus on a Tuesday.
Right. I mean, logically, you are probably never going to use it. You have a calculator on your phone, but you keep it anyway, just letting it gather dust. Well, it turns out your brain cells do exactly that. But instead of algebra or geometry, they are hoarding embryonic blueprints.
It's a truly remarkable concept, and it really fundamentally shifts how we view the life cycle of a human cell.
Yeah.
So today, for our deep dive, we are looking at a 2026 paper published in Nature Neuroscience by Kabi and colleagues, and they essentially set out to map the hidden software that controls adult human brain cells.
And the mission here, what they actually found is that mature, fully grown cells are actively holding on to a profound memory of their developmental youth. So, okay, let's unpack this idea of this hidden software, right? Because, we usually think of DNA as our ultimate biological blueprint. Like, if you have the DNA, you have the code.
Right, the hardware.
Yeah, but this research is totally focused on the epigenetic layer. So for anyone listening who needs a refresher, what exactly makes epigenetics the software to the DNA's hardware?
So, think of your DNA as the total hardware available, like every single key on a massive piano.
Okay.
Every single cell in your body has the exact same keys. It's identical. Epigenetics, though, that is the sheet music.
Ah, I like that.
Yeah, it's the physical and chemical structures that are just layered directly on top of the DNA. And that sheet music dictates which keys are played, how loudly they're played, and when.
So if a cell needs to be a neuron, the epigenetic software basically plays the neuron chord.
Precisely. And if it needs to be an immune cell, it plays a completely different chord.
Got it. So to figure out how these adult brain cells are, hoarding their old embryonic sheet music, we have to talk about the tools these cartographers use to actually map it out. Because they didn't just sequence the DNA.
No, sequencing DNA is easy now. They had to map the invisible software sitting on top of it.
Right.
And they primarily relied on two incredibly intricate techniques. And they did this at the single cell level.
Which is mind-blowing.
It really is. So the first technique maps a concept called chromatin accessibility.
Okay, break that down for us.
Sure. So if you were to take the DNA from just one single cell and stretch it all the way out, it would be about 2 meters long.
2 meters from one microscopic cell.
Yeah. So to actually fit inside a microscopic nucleus, it has to be wound up incredibly tight, like super coiled into this structure called chromatin.
Okay.
And when a cell actually needs to read a specific gene, it has to physically loosen or, unwind that tiny section of chromatin.
So if a section of DNA is tightly spooled up, it's basically locked in a vault, like the cell just can't read it.
Correct. So the researchers used a technique called SNATAC-seq.
Oh, wow. Okay. SNATAC-seq.
Yeah, it stands for single-nucleus assay for transposase-accessible chromatin.
That is a serious mouthful. Let's not get bogged down in the acronym. What's the actual mechanism there?
Right. Let's focus on how it works. They use a specialized enzyme, a transpose. Now, in nature, a transposase is an enzyme that jumps around and inserts DNA. But researchers have, hijacked it.
Hijacked it how?
To act like a pair of smart molecular scissors. It slips into the nucleus, but it physically cannot access those tightly wound vaults we just talked about.
Because the chromatin is too dense.
Exactly. It can only tag and sequence the sections of DNA that are currently unwound, exposed, and actively being read.
Okay, I want to make this visual for everyone listening. Imagine your entire genome is just this sprawling multi-story library.
Oh, that's a good analogy.
Right. Most of the books are shoved tightly onto shelves in these dark, locked archives. This transposase enzyme from the SNATAC-seq, it's essentially a librarian walking through the building, just making a hyper-specific list of which books are currently lying physically open on the reading tables.
That captures the exact mechanism perfectly.
Yeah.
But simply knowing which books are open isn't really enough to understand the cell's true identity.
Because you don't know what they're reading the open book for.
Exactly. You also need to know what the reader is planning to do with those open books. And that brings us to the second technique. And this is really where the study pushes the boundaries of what we can do.
Okay.
They used a method called nano-CUT and tag, but they adapted it specifically for archival frozen human tissue.
Which I imagine is notoriously difficult to work with, right? If tissue has been frozen, water forms ice crystals, cells burst, the DNA starts to degrade.
Oh, it's a logistical nightmare. It is not like working with a fresh sample in a petri dish. That is why their adaptation of nano-CT is so vital.
So what does the nano-CT tag actually do?
They used highly specialized antibodies to seek out specific proteins while the DNA was still totally intact inside these individual nuclei. And specifically, they were looking at histones.
Histones. Okay, so if the DNA is a thread, the histones are the microscopic protein spools that the thread is wrapped around.
Spot on.
And those spools aren't just dead plastic. The cell chemically tags them.
They do. So Kabi and the team were simultaneously hunting for two very specific chemical tags placed on these histones, specifically H3K27AC and H3K27A3.
Okay, let's use a mental trick to lock those tags in because they are the absolute key to this whole discovery.
Definitely.
So if the open books on the library tables are the unwound DNA, these histone tags are like brightly colored sticky notes just slapped onto the pages. For H3K27AC, you want to remember AC means action.
AC means action. I like that.
Right. That is the active mark telling the cell, yes, read this right now. And then for H3K27E3, remember, me means mute.
Mute, exactly.
That is the repressive mark, effectively slamming the book shut.
And the real breakthrough here comes from combining all this data using something called trimodal clustering.
Meaning they looked at all three things at once.
Yeah. They analyzed the open DNA, the action marks, and the mute marks simultaneously across the entire genome. And they discovered something totally counterintuitive.
Which was?
If you just look at the open books, the chromatin accessibility, different types of brain cells actually look remarkably similar. The data highly correlates. But when you look at the action sticky notes, those H3K27 AC marks, the landscape completely changes. They are much better at discriminating cell-type specific regulatory activity.
Wow. So the sticky notes tell the real story of what the cell is actively doing. It completely changes how you look at the brain. Your cells are essentially making these real-time sticky note decisions that dictate your health right now.
Yeah, right this second.
Okay, so let's do a quick recap here for you listening. Our toolkit is SNAT-ACSEC to find the open DNA and nano-CT tag to find our action and mute sticky notes on the histones.
That's the baseline, yeah.
So before we move on, a quick question for you to think about. If you could place an action or mute sticky note on any habit in your own daily routine, what would it be? Just think about that.
I'd probably put a mute mark on my late night snacking.
Honestly, same. So now that we have our action and mute mapping tools, let's apply them to the actual human brain to see what secrets these cells are hiding. Where exactly did they point this massive microscope?
They aimed it at 60 tissue samples.
Yeah.
And these came from 20 post-mortem human donors.
Quite a range of donors, right?
Yeah, ranging from 34 to 74 years old. And they strategically targeted 3 distinct anatomical regions.
Which were?
The primary motor cortex, which is BA4, that handles your voluntary movement. The cerebellum, which handles complex coordination. And the cervical spinal cord.
So 60 tissue samples from 3 completely different regions. The sheer volume of data there must have been overwhelming. How do you even begin to categorize the individual cells?
Carefully. Very carefully. After highly stringent quality control to just filter out the damaged cells, they successfully isolated just over 108,000 individual nuclei.
108,626 to be exact.
Right. And they then fed this massive data set into clustering algorithms. And the algorithm sorted them into 16 distinct cell families by looking for what are basically biological name tags.
Biological name tags?
And essentially, yes, marker genes.
Okay, give me an example.
For example, if a cell was densely clustered with action sticky notes around a gene called SOX10, the algorithm knew instantly it was looking at the oligodendroglia lineage, the OLG lineage.
So SOX10 is the name tag for OLGs.
Exactly. If the name tag was PDGFRA, it was an OPC, a precursor cell. PLP1 meant it was a mature oligodendrocyte, or MOL. AQP4 was the name tag for astrocyte.
And AF1 was for microglia, right?
You got it. And then they used RBFOX3 for excitatory neurons and GAD1 for inhibitory neurons. They mapped everything.
Okay, but wait. Finding a name tag on a cell is one thing. Knowing who actually pinned the name tag there is another. Like, if SOX10 is the master identity gene for these oligodendrocytes, how do we know what controls the SOX10 gene itself? Where is the master volume dial?
And that leads to the enhancer hunt, which is arguably one of the most elegant experimental mechanisms detailed in the entire paper.
Okay, so an enhancer, just to clarify, is a region of DNA that doesn't actually code for anything itself.
Right, exactly. Instead, it loops around in 3-dimensional space to act as a remote volume knob for a completely different gene located far away.
Okay.
So the researchers mapped out what they suspected was a distal enhancer for that SOX10 gene.
But finding a suspected enhancer in the genome is like finding a specific light switch in a pitch-black, sprawling mansion. How do you definitively prove that this specific switch turns on the SOX10 light bulb without accidentally, I don't know, turning on the garage door?
They proved it using a modified CRISPR activation assay. A CRISPR assay.
Oh, I love this part. We usually hear about CRISPR as a tool that cuts DNA, like genetic scissors.
Right, but they didn't want to cut the DNA here. They wanted to test the switch. So they used a dead Cas9 protein. It's often written out as dCas9.
A dead CRISPR protein, how does it even function if it's dead?
It's conceptually brilliant. So the Cas9 protein relies on a piece of guide RNA, which acts like a GPS coordinate.
Okay.
The guide RNA navigates the massive genome, finds the exact suspected enhancer sequence, and it docks. But because the cutting mechanism is dead, the protein just parks there. It doesn't break the DNA strand at all.
So it's basically a molecular drone just hovering directly over the target site.
Exactly. And the researchers attached a payload to this drone.
What kind of payload?
A transcriptional coactivator called P300.
So dCas9 P300?
Yes. By driving this drone to the suspected enhancer in human OPCs, the P300 payload forcefully applies an action sticky note right to that specific piece of DNA.
Wow. So they artificially cranked the volume dial themselves.
And it completely worked. By targeting the suspected enhancer, they successfully increased the expression of the SOX10 gene.
Without turning on the garage door?
Without turning on the garage door. They checked the neighboring genes like EIF3L and AONKRD54, and they were untouched. They definitively proved that this distal piece of DNA was the physical remote control for cell identity.
The engineering required to navigate a dead protein to a specific genetic zip code and drop an activating payload is just mind-bending.
It's incredible precision.
All right, time for another 5-minute recap for you all. So far, our tools are SNAT-TAG-seq reading open DNA and nanocut-and-tag reading active or repressed histones. We mapped 108,000 nuclei across the motor cortex, cerebellum, and spinal cord. And we just proved the existence of a specific genetic volume dial, an enhancer, for the SOX10 gene using CRISPR technology.
That covers the map and the mechanisms, yep.
So a question for you to ponder. What do you think is more powerful in shaping who you are, the genes themselves or the hidden enhancers that actually control their volume?
That is a deep philosophical biology question right there.
Right. Okay. So zooming out from the SOX10 enhancer. SOX10 itself is a transcription factor. And these are the master switches that bind to DNA to turn cascading networks of other genes on or off.
Yes.
So if we look at the brain's support cells, the glia, like the astrocytes and the microglia, what do their entire networks of master switches actually look like?
This is where the geographic mapping of the brain yielded a highly unexpected result.
Oh!
When researchers looked at the neurons clustered very distinctly based on their brain region. A motor cortex neuron looked fundamentally different from a cerebellar neuron in its chromatin accessibility. Their software was entirely customized for their specific location.
Wait, I'm struggling with this. You're saying the neurons are highly specialized based on location, which makes total sense, but the support cells don't care where they are.
Essentially, yeah.
But why would the support cells be completely generic? If I have a highly specialized motor neuron, wouldn't the astrocyte supporting it need to be equally specialized? That just doesn't seem biologically efficient.
It seems counterintuitive, right? Until you consider plasticity. Glial chromatin states are remarkably region agnostic.
Meaning they don't care about their zip code.
Exactly. Rather than being hardwired for a specific area, their chromatin architecture remains incredibly plastic. They're deeply adaptable. They can shift their functional states to respond to local microenvironments, injuries, metabolic demands, regardless of their exact geographic location in the brain.
Okay, I see. So they are the ultimate biological utility players. They just keep their options open.
Precisely. And by mapping out the specific transcription factors that allow this plasticity, the team discovered brand new regulatory master switches.
Like what?
Well, for instance, in microglia, which are the brain's resident immune cells, they uncovered a transcription factor called FOXN2.
Now, we already know microglia have master switches. How is FOXN2 any different from what we already knew?
Well, we knew about FOXK1, right? Which regulates an inflammatory pathway called STAT1. We also knew about FOXO3, which controls neuroinflammation, and how microglia eat cellular debris, a process called phagocytosis. But FOXN2 showed massive regulatory power while having zero overlap with those known functions.
Wait, really? Zero overlap?
None. This means there is a completely distinct, entirely unknown regulatory role governing immune response in the brain that we just haven't fully understood yet.
Wow. Did they find others?
Oh, absolutely. In astrocytes, they mapped ZBTB20 and HMX1. In the OPCs, they mapped PRRX1, which regulates their quiescence or resting state, and PAX3, which shows their dorsal lineage contribution.
Okay, but the data revealed something else fascinating about these master switches, particularly regarding how much noise they make.
Oh, the expression versus influence dynamic, yes.
Right. The researchers found that expression level, meaning how much of the transcription factor the cell is actively producing, does not equal its regulatory strength.
No, it doesn't at all.
Let's look at ZBTB20 in astrocytes, mature oligodendrocytes, and OPCs. It was the absolute highest expressed transcription factor. But when they measured its actual regulatory strength, its ability to actually control other genes, it ranked terribly low.
It really highlights a critical distinction in biology between abundance and actual influence.
I picture this exactly like a loud middle manager in a corporate office.
Oh, that's perfect.
Right. They are talking the loudest, they're sending the most emails, scheduling the most meetings, they are highly expressed in the office environment. But when you look at who actually holds the authority to sign the checks and change company policy, their actual regulatory strength is basically zero.
Exactly. The real power might lie with a quiet executive who sends one email a week.
Yes. Okay, another quick summary here. So glial cells are highly adaptable across the brain, relying on complex networks of transcription factors where sheer volume doesn't always equal power.
Simple as that.
So my question for you listening, does it surprise you that the brain's support staff, the glia, are actually way more adaptable than the star players, the neurons?
It surprised the researchers, I can tell you that.
Right. So this immense adaptability of the glia brings us to the core, arguably the most thrilling discovery of the entire paper.
Oh, absolutely. This is the big one.
We have established that glial cells are highly adaptable, maintaining open plastic software. But just how far back does that adaptability go? Like, can they actively remember what they were before they even became adult brain cells?
And to really grasp the stakes of this question, we have to look at a clinical reality. High-grade pontine gliomas.
These are devastating, aggressively fast-growing brain tumors, right, that frequently strike children and young adults.
Yes. And for decades, oncologists and neurobiologists have wrestled with a central mystery. How do they start? Why does a mature, stable cell suddenly regress, go rogue, and just start wildly dividing?
And the answer, as Cobby and colleagues discovered, lies in epigenetic ghosts.
The mechanism they uncovered is termed epigenetic memory.
Okay.
When they narrowed their focus specifically to the human oligodendroglia, the OLGs, derived from the adult cervical spinal cord, they found a chromatin signature that, theoretically, absolutely should not be there. They found primed signatures at the HOX gene loci.
Okay, let's break that down exhaustively because this is the absolute crux of the whole deep dive. What exactly are HOX genes, and what does it mean for them to be primed?
Right, so HOX genes are the master architects of embryonic development.
Architects.
Yes. When a human is just a developing embryo, HOX genes act as the ultimate layout planners. They map out the head-to-tail axis of the body. They dictate where the limbs should grow, where the vertebrae should form, where the spinal cord should route. Their entire job is to build the organism from scratch.
Right. So in a fully mature 74-year-old adult spinal cord cell, those architectural genes should be locked away in the deepest, darkest vault of the genetic library. The building is already built. You don't need the foundation blueprints once the skyscraper has been open for 50 years.
Exactly. But they aren't locked away. The researchers discovered that in these adult spinal cord cells, the HOX genes exist in a primed state.
Which means biologically what?
It means there's a co-occurrence of our two sticky notes at the exact same physical location on the DNA.
Wait.
The action mark, H3K27AC, and the mute mark, H3K27E3, are sitting side by side on the embryonic HOX genes.
Wait, action and mute at the exact same time. That is a biological paradox. It's like pressing the gas pedal and the emergency brake simultaneously. Why on earth would the cell do that?
It places the gene into a state of suspended animation. It is a bivalent state.
Bivalent.
Right. The gene is technically silenced by the mute mark, which prevents the adult cell from trying to, grow a new limb. But the presence of the action mark keeps the chromatin physically accessible. It prevents the gene from being shoved into the dark vault.
Oh, wow.
It remains paused, ready to be instantly triggered.
Did they confirm this structurally?
They did. To confirm this, the team used an advanced 3D mapping technique called micro-C. And they found that the physical, three-dimensional looping architecture of the chromatin around these HOX genes in adult cells bore a stunning resemblance to the exact architecture found in early developmental embryos.
So the cell is actively expending energy to keep these embryonic blueprints paused but readily available on the table. Why? Why do adult spinal cord cells keep their developmental software installed?
It acts as an evolutionary double-edged sword.
Okay, what's the first edge?
The first edge is regeneration. The spinal cord is a highly vulnerable, totally critical pathway. By keeping these Hox family genes primed, if a massive spinal cord injury occurs, these local glial cells don't have to start from scratch. They can rapidly drop the mute sticky note, unpause the Hox genes, and initiate immediate complex tissue repair using the original developmental blueprints.
It's an emergency repair protocol. That makes perfect sense. But you said double-edged. There's a dark side to leaving those blueprints lying around.
A profoundly dark side. Gliomagenesis.
Tumor formation.
Yes. This exact same primed state, this suspended animation, makes these specific spinal cord cells uniquely vulnerable to being hijacked.
Hijacked by what?
If a genetic mutation occurs, specifically the H3.3K27M mutation, which is a very well-known driver of severe tumors, it easily exploits this physically open, primed architecture. The mutant protein essentially hijacks the pause software and forces it to hit play.
Oh my God.
In fact, the chromatin architecture the researchers mapped in these healthy adult spinal cord cells bears a strikingly strong resemblance to the chromatin architecture of high-grade pontine glioma.
Let me give a visual for this dynamic because it is just incredible. Imagine a healthy spinal cord cell is a retired master architect.
Okay.
They are not actively building a skyscraper today. They're just managing the property. But they still have the old original blueprints unrolled on their desk just in case.
Right, the primed state.
Exactly. If an earthquake hits representing a spinal cord injury, they can instantly read those blueprints and expertly direct the emergency repair crews. But if a bad actor representing that H3.3K27 mutation breaks into the office and steals those readily available blueprints, they don't build a repair, they build a tumor.
That captures the duality perfectly. This epigenetic memory is a latent program intentionally persisting into adulthood. It is simultaneously our greatest biological asset for miraculous healing and a profound vulnerability for tragic cancer.
Okay, final structured recap for everyone. Adult spinal cord OLGs retain an epigenetic memory of their embryonic HOX genes. And this primed memory aids in fast tissue regeneration but risks triggering gliomas.
That's the core of it.
So my last question for you. If your cells can remember their embryonic state, what other hidden memories do you think are locked inside your DNA?
It really makes you wonder what else is paused in there.
Right. So, synthesizing all of this, from the transposius librarians reading the unwound DNA, to the sticky notes revealing glial adaptability, to the actual ghosts of our embryonic development resting in our spinal cords. What are the broader clinical implications of knowing our cells operate this way? Why should we care?
Because it fundamentally reframes our entire approach to neurobiology. We are no longer just looking for broken hardware or mutated genes. We are looking at misread software. We are looking at latent programs that still exist in the adult body.
That is not a hardware issue, it's a software glitch.
Exactly. Take a disease like multiple sclerosis, where the protective myelin around nerves is degraded. Or consider devastating spinal cord injuries. The cells of those areas already have the blueprints to fix the damage. The regenerative software is fully installed, and it's paused.
The sticky notes are just in the wrong place.
Yes. The clinical challenge moving forward isn't necessarily inventing a brand new drug to force regeneration. It is learning how to securely press play on the software that is already there without accidentally triggering a tumor.
We now know that developmental epigenetic memory persists in adult glia in the spinal cord, basically acting as a latent program waiting in the wings. If we can master mapping this memory, could we one day intentionally reboot it in other parts of the brain? Like, could we target the cortex to reverse conditions like Alzheimer's, or stimulate the spinal cord to completely reverse paralysis simply by developing therapies that rearrange those invisible sticky notes on our DNA?
It's the frontier of modern medicine.
Keep that in mind next time you think your brain is fixed in its ways, or, the next time you find an old notebook you just couldn't bring yourself to throw away.